Our new keynote speaker, Rui Costa. So Rui did his undergraduate study at the Technical University of Lisbon in Portugal, then went to UCLA in California to, from beach to beach, basically. Yeah. And if you've been to Lisbon, it's a beautiful beach in Europe, and California probably have a similar beautiful beach. So he did a wonderful graduate study with Arsenio Silva at UCLA. Then he went to Duke, did his postdoc. That's the place I met him when I was at Duke. So he did a postdoc with Miguel Nicoleles at Duke University. Then he started in his own lab at National Institute. I also have trouble to remember this. National Institute of Alcohol Abuse and Alcoholism, so NIAAA. And his main focus was on understanding the neurocircuits, neurocircuit basis of behavior related to the basal ganglia function. So he moved to Portugal in 2009, a wonderful place in Lisbon, a new institute called the Champalimaud Neuroscience Program, or what's the real name? The Unknown.
Center for the Unknown.
Whatever. Yeah. It's a wonderful program, funded, privately funded, and they have attracted many very good people over there. And one thing I benefit a lot from my multiple visits to Portugal is I still have several of really good graduate students actually from. They did most of some of the most important works in my lab, and so they continue to Rui not only trained very well, but also contribute a lot to the lab they joined. Then they return to Portugal and make wonderful discoveries. So you will hear a story. I guarantee you will be very excited. And I have read many of his recent papers. Welcome, Rui.
Thank you. Let me just connect these. Can you hear me? There you are. Good. So it's a pleasure to be here. Of course, we didn't plan that I would be the keynote speaker, but I hope not to disappoint you too much. So what I'm going to talk to you about today is about the mechanisms by which we can generate and shape novel action sequences. So if we look at an individual like this one here, you can see there's a variety of movements, very beautiful, very unique, that this individual can do that I cannot do. Each of us is able to build throughout life a repertoire of actions that is very unique. So if these actions would be innate, we would know, okay, there's something that is put in the genome from generation to generation that then builds a circuit, and that circuit then generates this behavior innately in the next generation. But what happens for things like this, that we build anew throughout life, right? If I'm born and I'm a programmer in Shanghai, certainly I don't need to have the same things as if I am born in Portugal and I'm going to be a farmer. So what do you put in the genome or in the organism to be able to do something like this? And most of the studies I'll tell you today, in fact all, will not be about this species, but about this species. And this is a mouse that, without no apparent rule, except that every time it presses 8 times in that lever, it gets a drop of sucrose here, has learned to do this behavior very well. Although the behavior, as you can see, is not innate. Mice don't come prepared to press levers 8 times, right? So what happens? So we've been looking at this in the framework of, you could say, well, trial and error, right? But what is it, really, trial and error? So trial is about trying, right? So generating variability, exploring. If you'd be talking about songbird, which we will hear during this meeting, it would be about babbling to the world. But this, you could say, is motor babbling, right? When we're born as kids, we don't know exactly many of the things we do, and we just generate to the world. And then you'd put a mechanism of selection. You grab what works, and you repeat it, right? Again and again. This is maybe oversimplified, as there are many mechanisms, for example, that one can construct of selection, but this is the framework that I want to talk to you about, right? So let's say you just need to put two things in the organism to be overly simplistic to learn new stuff. And so what we'll talk a bit about today is how are new, novel, self-paced movements or actions generated? How are specific actions then selected? So if you do a lot of stuff, I have to do something to compensate for the absence of Carl Disserot, so I'll do a lot of gesticulation in the podium. So imagine you do something and you hit this, right? And then you want to do it again, right? You can repeat it. How are those action selections? And finally, how are they automatized into, let's call them behavioral units or modules or chunks? So what's going on in the brain? Okay, good. So if we'd be talking about an inanimate object, like a red ball there, right? How do you generate any behavior out of it? So one of the great things about this symposium, or at least it was last time we were here, and I hope it's now, it's the student participated a lot, so I hope there's lots of questions. But basically you have to you apply a force here, right? And if you apply a force, the ball starts moving. And then note that if nothing else happens, the ball continues to move, right? And you need to apply another force if you want to stop it or change trajectories, right? So this is our framework of how you generate movement out of nothing in something that is not a living organism. But so can we take anything from here, right? Well, where's the force that makes you start doing something, right? Especially if we're talking about self-paced actions, where there's not a particular stimulus predicting what's going to do it. And then can we take anything from these? So if you notice, one of the things is the ball, once it's in a trajectory, if you compare the current time to past future, right, the past can predict the future. Basically, there's some predictability there. So the behavior, we could say, is the same. But there are points where the past and the future are not the same. These points of transitions, the ball starts, the ball stops, changes trajectory. Can we apply these to the study of animal behavior? Here's some work by Alex Gomez-Marin, who comes from physics, looking at the movement of an animal just in a box without any stimuli. And this is a comparison of the past and the future of that animal. And you see at some points, the past and the future, time zero is now. The past and the future at some points are very symmetric, right? And at some points, they are very asymmetric, right? Now they're very symmetric, the animal's locomoting forward, then it does something, you know, like a twitch, you see some asymmetry developing, passing through. And here is the same thing, but just looking at the asymmetry. So if the animal is in a flat line, it's either stopped or within the same behavior. But if you see these peaks of changes, right, these represent transition points, right? So, of course, this framework is still being developed, but the easiest thing to ask is at the moment where the red ball starts, right? That we could determine pretty independently. So, what happens in the brain when there's a self-paced transition from being immobile to moving? And I'm just going to introduce you here briefly. This is a lot of the brain of a mouse, and throughout many parts of the brain course these cortico-basal ganglia thalamic loops. Basically, the cortex sends glutamatergic input, and the thalamus to the main nucleus of the main input nucleus of the basal ganglia, which is the striatum, that then sends out inhibitory GABAergic projection pathways. And if it sends out directly to the output nucleus, which in itself is inhibitory, we call that the direct pathway because it reaches it directly. And if it reaches it indirectly, it was called the indirect pathway. So anyway, if you record from several points on this loop, let's say M1, M2, the striatum, you see neurons with activity prior to the movement, but they are specific for the action somehow that's going to happen. But if you record from dopaminergic neurons in the lateral part of the substantia nigra compacta, and you align them to the moment of initiation of a movement, this is what you see. Right? So you see a very sharp burst of activity in these neurons. And of course we know from our work and that of many other people, that if you have excessive dopamine, you're hyperactive or psychotic. If you have very little dopamine, you have Parkinson's. And basically, if you have a lot of dopamine also in mice, you have a lot of transitions of behavior, here measured by EMG activity. If you have no dopamine, you have a flat line. Okay. But this doesn't help us really ask if dopamine, the dysphasic transition or phasic transient activity of dopaminergic neurons is necessary or involved in the transitions. For that, we need better temporal resolution. If an animal is dopamine depleted and just staying there, okay, then it can't move. So what Joaquin in the lab started doing is using ArchT expressed specifically in the dopaminergic neurons of the substantia nigra pars compacta to transiently inhibit the neurons for 5 seconds, say. And at the same time, following the behavior of the animals with video and accelerometers in the head. So what you can see here is that, obviously, if an animal is moving, and this is the case of the YFP here in black, and continuing to move, you see no effect of light. But if you apply light in animals expressing ArchT, you see a decrease in movement that is pretty sharp. Okay? And if you look at what the distribution of movement is in general, you see it's bimodal, right? So there's this point here, this little like low peak, which is basically moments of immobility, what we call immobility by looking at the video, but there's still some small postural adjustments, and this is overt movement. So you see a shift from to the left, and what you can see here is just comparing acceleration and video, basically saying you see the same thing. Okay, but this is not the question. So you transiently inhibit dopaminergic neurons, you move less, right? But let's now align the trials to when the animal is moving versus not moving. This is what we wanted, right? So if an animal, for the 300 milliseconds just before the light came on, was immobile, so was in this case here, what you can see is you inhibit light, there's no movement, right? If you don't apply the light, the animal will start moving. So if the animal is not moving for 300 milliseconds before ArchT is activated, the animal will not move for many seconds. So we'll have a difficulty initiating movement. But look, if the animal for those 300 milliseconds is in overt movement, nothing happens. It continues to move. Well, you could say it trickles a bit down, right? So, well, maybe the animals are becoming slower, bradykinetic, or maybe they stop, and after they stop, they have difficulty initiating again. And the situation is exactly this second situation, right? So what you can see is that when the animals stop, right, they have difficulty initiating movement again. So there's no difference in stopping, but there's a difference in initiating movement again. I don't know if you can see that. Okay, fine. And if something, when you look at animals that are moving, the latency to stop, or the amount of time it takes to stop, is now increased, so the bouts are longer. So, in a sense, it's almost like if they are not moving, they cannot transition into movement. If they are moving, they have difficulties now stopping. Okay. Now, I want to show you here how one of these neurons really behaves in vivo. This is a photo-identified neuron. It's a neuron in the SNC, a dopaminergic Th-positive neuron, where we express channel rhodopsin, and the neuron responds. And now here, it's aligned to the point of initiation of the movement. And you see, it does a small burst, like, right? 3-4 spikes, 80 milliseconds, 70 milliseconds apart. So if we mimic these in vivo, well, mimic. If we apply the same pattern in vivo, so let's say 4 pulses, each pulse 10 milliseconds at 14 hertz. It's not exactly mimicry because we're activating many neurons synchronously and we don't know how synchronous they are. But what you can see is this is sufficient to cause a transition to movement. Just this brief pulse of activity. If the animal is not moving, it transitions into movement, and it stays like that for a while. So it's not just like a startle, right? So it stays moving for a few seconds. If the animal is already moving, if something, it stops moving, right? So, which is what you can see here. There's a small blimp. It's hard to study the stopping really in this situation or the transition. So we did the same experiment, but now in a running wheel where animals really are running and the only behavior they do when they are not running is stopping. And we see here the instantaneous firing rate of a dopaminergic neuron just before the animal started to running and before stopping. And now we're going to do the same thing and apply a brief stimulation during the run, and you'll see what the animal does. Right? So this square here was when it was stimulated. We'll repeat once again. You'll see it coming up here. This is accelerated, obviously, but the animal stops for a little while and then continues. So what am I telling you? That dopaminergic neurons are phasically active during the initiation, generation of self-paced actions, or transition between them, right? Whatever you'd want. That activity in these neurons seems to be necessary for those transitions, and that the small burst, the small transient activity, is sufficient to cause the transitions. And you'd say, well, that's great, right? Dopamine and movement, but we know a lot about dopamine and reward and error prediction errors. So how is this compatible? So maybe it is not compatible, right? Maybe these are different types of signals or neurons, but maybe it is compatible. So what we know from the work of Wolfram, Schultz, and then others is that dopamine neurons encode error predictions. So if an unexpected, unpredicted stimulus arrives in the world, dopaminergic neurons would fire, right? And then if something good happens, they would fire more to the unpredictable stimulus. If it's predicted, you don't fire. So how would we translate that to actions, right? So imagine you transition between an action that's the same action to something new, right? At this point, maybe this is self-paced, right? But maybe you signal using the same permissive signal that is necessary for the transition. Maybe you send to the whole brain a signal saying this is a novel action. And it's unpredicted because in this past future framework that we're talking about, right, this is a transition that's new, right? It's not what's expected from the ongoing movement. And if you think about, so which actions would you reward if you get something good? You should back up, trace to the last novel thing you did, right? Because that's where most likely the cause of that outcome is. Okay, but this is a lot of blah, 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 but if this is true, then the neurons that respond to reward, maybe they should respond to movement. So I'll show you here just a little bit of preliminary data with just not a lot of analysis, but of a mouse performing an operant task like I showed you at the beginning. I apologize for the resolution, but I don't think it's the screen here. Something in the translation. But I don't know if you can see here the mouse moving and the aligned video with this small inscopic system imaging 7 dopaminergic neurons here. They're a bit out of focus, but you can see them there better. And I can show the video to people at the end if you want. And you can see here when the animal enters and licks reward, many of the neurons fire a lot. So this is this moment here. And you see this neuron is this particular one. You see it just fired to a lick. But then when the animals initiate the movement again, you see 1, 2, 3, 4, 5 neurons show a transient aligned to this point here to just go and collect a reward. So, of course, we're doing many more animals. So far, we have just a few. But it seems consistent that although not every neuron that responds to the delivery of reward or unexpected reward responds to during movement initiation, many of them do. And the other thing I would like to point out is how similar the activity between the different neurons is. So we do think that this transient activity of dopaminergic neurons is related to behavioral transitions, and maybe also the same, brain transitions, brain state transitions. Okay, but if this is true, then very well-trained actions, so if you overtrained an action, then this action becomes like a habit, like a stimulus response, becomes very automatized. So you start it and you do it till the end. So it should be less, dopamine should be less involved in this type of actions. Can we test this? So here is an animal, and this animal is, all the receptors of dopamine have been blocked. This is an experiment I still did some time ago. And I'm going to place the animal in a rotor rod, the accelerating rotor rod. It stays like this. There's no self-paced movement. Then I initiate the rotor rod. And you see I even have a reflex here trying to grab the animal. The animal falls. There's no self-paced movement. You see it falls with the 4 paws down. If you place it on a beaker of water, it swims. So reflexive movements are intact, but no movement. What happened if I would have trained this animal for 2 weeks before in the rotor rod? Every day, 10 trials, let's go, right? This is what happens. So still no self-paced movement. I deplete now. I place the animal in the rod, still complete immobility. I start the rod, and now the animal moves. So something happened from here to here, just with training, that rendered the same kind of behavior now less dependent on dopamine, right? And if you see this movement, it's good, the video slow down a bit, it's almost triggered when the animal is about to fall. So what happened, and I'll summarize a lot of work here, is that we found that just this training caused plasticity in the inputs from motor and somatosensory cortices to the part of the striatum that's called sensory-motor striatum, or the putamen, if you will, that receives that input, such that now, if you look even wholesale, the threshold for spiking has been decreased, and you don't need dopamine so much. the threshold for spiking here in the striatum. The other thing we saw was if we measure the activity in M1 and dorsolateral striatum using multi-electrode arrays in vivo and looked at the trial-to-trial variability, this variability was very high early on, right, in the first day of training, but it decreased with training till it was very stable. So what do these two things mean? So one is a phenomena that resembles something almost like LTP, but it's like it occurs in vivo naturally, happens here where the glutamatergic synapses get potentiated. At the same time, the variability in neural activity, which is very high the first time you're trying, or the mouse is trying to run, the trial-to-trial variability, becomes consolidated into one type, and from trial to trial now you activate always the same neurons in the same way. So this raises the hypothesis that plasticity in basal ganglia circuits, so onto the striatum, is a potential mechanism for selection, the action patterns and the neural patterns that work. Right? So does this make sense? So you try many things early on, and then at one point you just stick to one thing. The movement-to-movement variability is low, but the neural activity, trial-to-trial variability, is also low. So can we test this? To do that, and that the striatum is important for that. To do that, we used a different type of task, which is the one I showed you in the first slide, that video. So basically, an animal is placed in a box, there's a lever there, and if the animal presses this lever 8 times, a drop of sucrose is then released in that magazine. Now, these are the only rules in the world of that animal. The animal starts by pressing and exploring, and then eventually pressing more. And early in training, if you look, this is a whole session, right? You have here the presses are these little circuits, the lever presses, and every one of these black marks is an entry into that food port or magazine. Every one of the red bars is a leak. So you see the animal starts pressing a few times before it goes to leak. But you know, it's the animal, every 8 press gets a reward, but it could press 1 + 1 + 1 until 8. It could press 16 and get 2. It could press 6 + 2. There's nothing in the world really saying, hey, you should press 8 times. But look, just a few days later, 6 to 12 days later, what you see is that the behavior becomes very organized. So let's see that they're trying to focus up there, but it's, can you see that? That the behavior is very organized. So we can quantify these. And basically what you see is that they press about 8 times with training. But this is not so relevant. Some animals press 5, some press 9. We know that they cannot count with this amount of training, count 8, 9. You know, they can distinguish 4 and 5. They do so faster, so the sequence duration and the within-sequence press rate decrease, the inter-sequence interval decreases. But this is what I want to show you, to draw your attention to. The coefficient of variation of the behavior, so how similar the behavior is in each animal from trial to trial, or from every time it does an attempt, a sequence, decreases for all the measures, right? So these indicates that something is being selected, and that animal then does the same thing over and over again. Okay, is plasticity onto the striatum at the glutamatergic synapse is necessary for this? And to test that, what we did is we took advantage of a Cree line that just postnatally deletes from the spiny projection neurons that constitute 95% of the striatum and that receive those inputs. And we deleted NR1, the essential subunit of the NMDA receptor, from those neurons. And those neurons then become void or with very little NMDA. And basically, this is important. This disrupts LTP. and a bit bursting of these neurons. And what you can see in these animals is this. Look, they can press. This is the behavior early on. This is the behavior later on. They can press, but the behavior never seems to get organized. And if you see here, just comparing the coefficients of variation of the mutant mice in red and the control litter mates in blue from early to late training, what you can see is it decreases in the control litter mates. It stays flat in the mutants. So this is evidence that maybe the plasticity at those synapses, at those inputs in striatum, is necessary for the process of selection. Not so much of doing, but for selecting a particular pattern. Okay, but this is a very easy task, you know, like this is not what we do in everyday life. So what if we train animals to do something very hard where they just need to optimize one of the features of their behavior, right? That's more the everyday life. So what we did is now we trained the animals. They just need to press 4 times or more, but at increasingly faster speeds until they need to press at about 6 hertz or 8 hertz, right? Till they need to go very fast. Now, they can press more than 4, but at least 4, but the frequency at which they press it needs to increase. Now, we look not at coefficient of variation, but Fano factor, which is another measure of within animal variability. And you see for frequency, which is the task-relevant domain, it decreases. For the other irrelevant domain, it increases. So, you know, how many presses it does, if it takes a long time to get to those 3 or 4 interpress intervals with very high frequency, it doesn't matter. And if it takes many presses either. So the variability in the other domains actually increases. Now, if we record activity, again, in the motor cortex in layer 5 and in the sensory motor striatum, where it projects to, and we do the same measure, the Fano factor of the activity, and now of the same neurons, so Fernando recorded continuously from about 12 hours before training without unplugging during 4 days, and about 30% of the cells we could keep stable. And if you do that, you see the variability, the Fano factor of the activity of these neurons is very high early on, then it decreases. So this is training down here. But if you take from another period of baseline in the task, it's not that these neurons just became more variable, it's really during the movement. If you look at the firing rate, either during the period they are pressing or during the baseline, the firing rate is constant, right? Doesn't change. So it's really how variable it is. And this is the same data just depicted in a different way, showing all the neurons that are stable. And you see red colors here, meaning very variable early on. And you see almost all the neurons show the same pattern in striatum and motor cortex. But for firing rate modulation, not much. So the firing rate seems to be relatively constant, but it's almost like if the neurons are trying different patterns or the ensembles of neurons. Okay. At the same time, the variability in M1 and striatum, not the activity, but the change in activity, the Fano factors, become coupled, right, with training. And now if we go and correlate the activity with the behavioral features, you see there's an increase in coupling or correlation, but just for frequency, not for the other domains. So somehow the change, the reduction in variability of these neural patterns, the consolidation of a neural pattern, correlates with the consolidation of a frequency pattern, but not the other domains. And again, if we use the same mutant here and delete the NMDA receptors from medium spiny neurons, what you can see here for frequency is the control litter mates decrease the variability, not the mutants, and for the other domains, there's no difference. So this really leads to the hypothesis that, wait a minute, basically, so you can do something, and if that's what's relevant to get an outcome, you select that. But we know we can't select specifically the muscle patterns, right? I mean, imagine I do something like this, and you say, oh, you just got 50 yen, you can go to the bar, but do it again, right? I can't. I would have to select the neural patterns that led to that behavior. Okay, so can we do that experiment directly? So what we did is now, we are recording from M1 of these mice, and now what we do is, we take a neural pattern, and we say, when these 3 neurons increase firing rate and other neurons decrease firing rate, you drive an auditory cursor up in pitch. And if the opposite happens, you drive it down. So early on, you get a lot of beep, boop, 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 beep, boop, boop, boop. But that auditory cursor is giving feedback to the animal about its own neural activity. And one specific pattern leads to reward. Okay, can the animal select that pattern? And with time, what you can see here is, yes, they learn to select the neural pattern. And I mean, we did a lot of controls, irrespective of movement, right? We even anesthetized. This is in the area that controls whisking. We even anesthetized whisking. So this is chance, and you see it gets well above chance. Okay, we use the same mutants again. We delete the NMDA receptors, and they cannot select that neural pattern. So we take this second part then about selection to say, okay, it is true that variability in neural and behavioral patterns decreases with training, as if something is being selected, and that specific patterns seem to be selected and reinforced, and that plasticity in inputs to the striatum is necessary for this selection. Okay, fine. But I showed you, or I told you, that every animal does everything slightly different. The press, it's just to itself that it gets. So what is being selected? What are each individual neuron doing in the basal ganglia, right? So you select for one this, for another that. What is it? And so if you, I mean, in this particular task, the animals do organize their behavior as bouts of presses, right? And so these could be like little modules. But if these are modules that the animals, so meaning if one module is now a few presses and not just a single press, right? If the animal has selected these, then you'd need to have some neurons that are just correlated with the beginning and end of that module, and some neurons maybe that treat that module as if it's one action. And so we observed that. So these are recordings of medium spiny neurons in the basal ganglia. These here are in striatum, but we find them in other parts too. And you see here neurons that are phasically active just before the first press of a sequence of presses, but not in the middle, and neurons that are phasically active at the end. And we also see neurons that are active sustainedly throughout the whole execution, so all the movements, and neurons that are inhibited throughout the whole execution. So it seems that what's being selected is related to the organization of the behavior of the animal into modules, automatized modules. Okay, now these recordings were made here, but I told you in the beginning there are two types of projection pathways. In terms of neurons, if you look at them, they're almost indistinguishable, but there are different predictions. One is that the neurons of the direct pathway, these inhibit thalamocortical inputs, so minus, minus, and so-called, this pathway was also called the go pathway. And neurons from the indirect pathway, they over-inhibit the thalamicocortical projections. So we went to look, maybe these neurons belong to different, these type of neurons organizing the parsing and the concatenation of these modules belong to different pathways here. And here's the data from photo-identified neurons, and what you can see is, and I forgot to tell you one thing, we have the advantage that these neurons express D1 type receptors, these express D2 or A2A, and so we can photo-identify them or image them. And what you can see is the similar proportion of neurons is active early on, right? Before the start or at the end of the stop of both direct and indirect pathways. However, during the execution, or these neurons that are more related to treating a whole sequence of presses as one single action, they were the neurons with sustained activity, they're more expressed in the direct pathway, and the neurons with inhibitor activity more in the indirect pathway. So finally, we ask the question, is activity in these basal ganglia neurons then necessary for the initiation and performance? So now we test it, right? So, and very briefly, we can place an infrared beam in a task like what you showed, and when the animal goes from the magazine to press the lever, we can put a flash of light, for example, and inhibit the neurons using Arch-T. And what you can see is that when you inhibit the neurons, the latency to start basically increases. So the animals take longer to start. However, in both pathways, so you could say, well, then they're both necessary. Yes, but for different reasons. And here is now a decomposition more of these. So in the D1 Cree animals, if you look at what they do in this zone between pressing and checking, or if you look at the videos from top, with light on and light off, there's no difference. They just freeze and then initiate. The D2 neurons, when you inhibit them, the animal switches to do something else. So this maybe indicates that maybe this is not so much, the model is not so much a go, no-go in terms of hyperkinetic, hypokinetic of the direct and indirect pathway. But maybe they're both coactive in the beginning, but the direct pathway is necessary to start the wanted movement, and the indirect pathway to inhibit competing movements. So if you inhibit both, you have difficulty initiating, but one you can't do, but you don't do anything. The other you can't do, but you do everything else except for that. So if this is true, then during the behavior, if we would stimulate the direct pathway, we should see animals continuing to press, and if we stimulate the indirect pathway, we should see animals switching, and this is what we see here. So now, this is an animal with no light and with light, the same animal, and you see when the blue light is there is when we're stimulating, and you'll see that as long as we're stimulating, the animal continues to press. So the sequence length becomes much higher. And this is only after training. So in naive animals or with very little training, it doesn't happen. And the opposite, for D2 stimulation, you'll see when we stimulate, the animal switches to go do something else. So we do think that something akin to this model is perhaps more the way that these modules are organized in the basal ganglia. Okay, but now, as the 40 minutes are up, I'll just tell you that, what did I tell you, in case it was very boring. So what I tried to tell you is that dopamine, phasic dopamine signals seem to be critical for the generation of self-paced movement, for the initiation of movements or for the transitions, if you will. And that the execution of these well-trained movements then becomes less dopamine dependent. So if you continue to train and the things become less self-paced, they become less dopamine dependent. Then that of the things you generate, you may select a few that lead you to specific outcomes. And you do that by selecting the neural and behavioral patterns that interest you and that plasticity in the basal ganglia, So specifically, the glutamatergic inputs to medium spiny neurons seems to be critical for this. And then finally, that what is being selected then and organized are behavioral units. I mean, you could think of the same process just for thought, as thought units, and that are automatized, organized in chunks, as was proposed by Lashley early on, and that the basal ganglia subcircuits, the different cell types, seem to have a slightly different role in this organization. And finally, I just want to thank the people in the lab that actually do the work. This particular work that I taught today, Joaquin, Fernando, Alex, and Lauren contributed to, and some people that now have their own labs, Shinjin, Fatwell, and Guhong. Thank you very much for your attention. So we're staying here until the first student asks a question, so it may take a long time. There we go. Hey.
Yeah, you showed that the transient activation of the dopaminergic neuron can only affect the initiation of the movement, but not the ongoing movement. So the question is, what's the mechanism to gating the different the behavior at a different movement state?
Yes. So the activation, the activation which is here does affect the ongoing movement there, right? So it causes if something has stopped, but the inhibition doesn't, right? So this is, perhaps I misspoke. So the inhibition, this is what you mean. The inhibition, after the movement has started, it doesn't make it stop. If something, it now has difficulty stopping. So what else is controlling the parameters there? Is that what you're asking? So, okay, I don't have all the data, but I'll try to be very brief and tell you what we think. So if we measure activity in M2, in striatum per se, in substantia nigra reticulata, we see a lot of neurons with that are related to the initiation of the specific action, and then that activity actually, Anatole, I don't know if he's going to talk about it, but correlated with, for example, the speed of the ongoing action, right? So the transient activity of the dopaminergic neurons, we see it more as a permissive signal for transitions. And so what this would imply is that when you start moving, it's still the same movement or movement state. You can accelerate it, decelerate it to a certain extent, as long as you don't change it a lot. If you change it a lot by stopping or changing orientation, then it's when I think dopamine would be necessary. But the rest, I mean, the only evidence we know from manipulations is a bit the parvalbumin interneurons in the striatum can modulate a bit the speed of movement, and the chat cells as well, but not...
Training will lead to dopamine less dependent in the basal ganglia. By the way, my question is, for the Parkinson's disease patient, they have problem for locomotion. Locomotion seems to be very well trained. So, can you comment on this, please?
Yes. Can I answer and at the same time open a video of a Parkinson's patient? Probably many of you have seen it, but I don't know if the students have seen it. And I think it will be very, very interesting. Let me see here. Sorry. I think it may be on this one, but I'll try to just see. So basically, I hope to show you a video of a Parkinson's patient that lives in Holland and has ridden a bicycle all its life. And then what happens to the movement in the absence of any dopaminergic treatment. But locomotion in humans is very interesting. I don't know if you guys followed this paper by Laconiti, trying to measure the primitives of movement. And so when baby humans are born, when we are born, we have basically two components for walking. And those components are the same in the human neonate or the rat neonate. Then when we are toddlers, we change to 4 components, and those 4 components are similar between the human toddler, the adult rat, the adult cat, the adult monkey, guinea pig. And then the adult human develops 2 new components, and these 2 new components are, hopefully this is it. These 2 new components are indeed learned. And this is very puzzling. So our locomotion, which we do so much as adults, is something very learned and very much under like self-paced control. This sounds strange, but if you know me, for example, you know I'm incapable of keeping a deep conversation and continuing to walk, which frustrates a lot of my friends. So if you say, oh, Rui, but really what was the I stop immediately, right? But anyway, so my thing is that human adult locomotion is really very hard to automatize for a long period of time. Somehow, we're always keeping it self-paced. Oh, this is not the one. Anyway, I'll find the video and show you guys later if it doesn't show up here. But the video is of a Parkinson's patient, as I said, starting to walk and having difficulties, and then put on a bicycle. But I'll keep the other question and then show the video in a moment. Sorry, please.
Okay, thank you. You just showed that the dopamine neurons in the striatum has facetic activity during the action, and the function of striatum is also strongly regulated by other neurotransmitter system and GABAergic and the cholinergic. Have you ever tested the other? And maybe they also involved in the different phase?
Yes, so this is interesting. We tested a bit the cholinergic. We didn't test directly the serotonergic, but data from other people, and I think other people here may be commenting, also seems to be involved in movement. They didn't do the analysis that we did, separating from when the movement already starts or not. I should be careful and not call it movement. I should say transition to movement, because there's nothing really, right? I mean, if it's not a muscular twitch or something. So we tested the cholinergic interneurons, and there you could see that if you overactivate before the initiation, you may have a little difficulty or slower initiation, but nothing else. So this, obviously, there could be more sophisticated analysis that we need to do, but there, if something, the permissive signal that allows for that action to be, let's say, selected, or this is the one I'm going to do, seems to be like coincident. Imagine from the cortex and thalamus coming into striatum glutamatergic inputs, and if there's a transient dopamine at the time, it's easier to select. Of course, if you trained a lot, then it's... So we think at least the cholinergic and the parvalbumin neurons that we tested are not necessary for that initial phase. But we don't have as good experiments there.
Okay, thank you.
So this is the video, if I can show.
As to the start cells and the stop cells you recorded, are there any difference in the projection from SC to the striatum? Do they belong to the direct pathway and indirect pathway?
So the start and stop seem to be equally in the direct and indirect. But now what we're looking at is from SNR downstream. SNR has very diverse projections. Are these, although they are, so being from the direct pathway just means you project directly from the striatum to the nigra, right? And being from the indirect, it just means you project to the pallidum. So we look at the activity of these neurons. We cannot find one that comes before the other, and the proportion of neurons active seems to be the same. However, what we started to do is the neurons that these neurons synapse onto may be very different. And so we started dissecting. I mean, the GPE people know that is very diverse. We started dissecting the substantia nigra reticulata, and it's very, very different. So you have neurons projecting to lateral superior colliculus that are different than the ones projecting to medial superior colliculus, to PPN. And so it could be that within what we call a homogeneous population, there are neurons targeting neurons that project to different brain areas and even that are different cell types. That's our current hypothesis.
Thank you.
Okay, so I'll show the video. This is a Parkinson's patient without medication, and as you can see, this is in Holland. It cannot initiate self-paced movement. If the assistant touches the foot, if it's stimulus response, it eventually can do it. But I mean, besides the tremor, like, of course, this is a rather advanced stage. So then you see, again, and sometimes then the problem is stopping, right? So now, this is hard to believe, but this is the same patient just walked outside the door of the hospital and put on a bicycle without medication, right? The first time I saw this, so we had just published those results, and a friend from Holland called me and said, "You have to see this." I opened the video, I was like, wow. But then I start panicking. I'm like, what's going to happen now, right? This is like, seems that the road gets to an end. But this is pretty impressive, right? Like, and I think if something, it alerts that it's even more complex than what I thought, right? Because you say, okay, this is automated. It's a trick, the rotorod and these, once you start, you keep on going. But look, it goes fine. And now there's something that to me is really amazing, right? The person brakes, right? So I don't know if you see. And then it jumps off, right? And then says, look, there's the camera, walk, walk. And then, like, again, the same problem. So there's something weird about locomotion, at least in this context, right? That's always self-paced. So these people that I know in La Coruña, in the north of Spain, they say, okay, if it's stimulus response, let's give them a metronome. Toc, 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 toc. And they walk, right? You stop the metronome. So, I mean, this is the extent of our ignorance at the moment. But it's interesting. Oh, one more. Sorry.
Yeah, you detected the role of dopamine neuron from SNC. Did you try to find out the role of dopamine neuron from VTA? I think there is something about a reward in the self-paced behavior.
Yes. So this is exactly. That's a great point, right? This is exactly what I was trying to say with that eligibility trace. So when we do something new, right? So it's because we're seeking, imagine self-paced behavior, you could argue, well, you wouldn't move if you don't expect anything better, right? So you should stay there forever. Why would you, I mean, if there's a stimulus or something, but if nothing happens, right? Well, maybe you fidget a bit, or we have a trigger, but you could say, no, every time we move, we expect something, right? And obviously, either you expect to avoid something bad or to get something good. And so these would be the transition points, right? And that was what I was trying to say with that eligibility trace thing, that the same neurons that would be necessary for the transition onto something. So it's like different stimuli, right? The dopamine would just encode if something is unpredictable and signaling something good or bad. But our own actions, right, maybe the part of the brain that's initiating them, like PPN, M2, right, then needs a signal that says, hey, you're doing it. Mark these, right? There's something new in case something good happens in the future.
How can you identify the dopamine neuron you activate or inhibit is from SNC, not from VTA?
Yeah, so this is a good question, right? So the really true, I mean, I'll give you all the lowdown. We can just say putative dopaminergic neurons because these are either TH positive Cre or DAT Cre, so we try to use more than one Cre line, so it's not too specific. Then we do have a line that's more VTA. We don't have a line that's more SNC. Well, we have one that's a little bit more SNC, but not a lot. So what we have to do is target the injection with low volumes and then count. So for every time, see which animals were just affected. So you can see the expression after the experiment. You can cut all the slices and count, and this is what we do. So maybe one last one. I don't know if anyone wants to.
Well, as you mentioned, NDA is important for the action variability. And can you guess this plasticity happens on what kind of cell types, like the dopamine-related synapse or the cortical striatal synapse? Thank you.
So it's an excellent question, right? We tested here mainly at the entry point, right? Now, if this is true, and to select, let me see where we have a scheme so I can answer pointing to, let's say here, right? If it's true, and imagine, right, you're selecting glutamatergic inputs here and here, it could be, well, that's the only place of plasticity. It's highly unlikely. I mean, at least in M1 we see some plasticity, but as you mentioned, the glutamatergic inputs to SNC, we've shown in a different study, are necessary for habit formation. So again, so I'm not going to say it's throughout the whole circuit, and then we have even projections out directly to the brainstem, which we think are controlling the movement, as I answered. But it's probably more than just a striatum. So you're right. I mean, it's hard to know the specificity. So is it if you delete in any synapse within this loop, would you get the same results? We don't know because we haven't done it, but it's a good question because it's very different. If there's something special about that specific synapse that is selecting, there's a huge dimensionality reduction going from here to here. Like many, many neurons to few. Or is it anywhere and you just cannot do it? And I think it's an excellent question. I can tell you here and here seem to be necessary and somewhere in the cortex, but we haven't tested everywhere else. Thank you.